This diagram illustrates a major biological challenge. We have a 2-meter-long thread of double helix DNA, and it has to shrink and coil down to fit inside a microscopic nucleus without becoming a tangled mess. The solution to this storage problem is the chromosome. Over the next few minutes, we will break down exactly how this packaging system is built, step by step, using the specific structural requirements of the board exam. Look at this checklist of chemical constituents. A chromosome requires 2 base ingredients. First, the nucleic acids, DNA and RNA. Second, it needs metallic ions, specifically calcium and magnesium, to stabilize the structure. Next, we add the structural proteins. These are split into 2 precise categories: histones, which are basic in nature, and non-histones, which are acidic. Note the distinct chemistry here. The histone's basicity is what allows it to interact with the acidic DNA. Mislabeling these 2 proteins as both being basic or both being acidic is a common error that ignores the fundamental chemical bond required for packaging. This split-screen diagram maps out the manufacturing pipeline. On the right, you can see the bare DNA strand wrapping tightly around a core of 8 histone proteins. This combined unit is called a nucleosome. As thousands of these nucleosomes link together, they coil into a dense, continuous spiral. We call this thick telephone cord-like structure the solenoid fiber. The solenoid fiber loops and folds over itself repeatedly to create a tangled mass known as chromatin. As the cell prepares to divide, this chromatin compacts even further into a dense, distinct rod called a chromatid. Finally, 2 identical chromatid rods join at the center. This joining creates the classic X-shaped structure we recognize as a fully formed chromosome. It is helpful to understand the functional difference here. Chromatin is the loose, accessible form of DNA used for everyday cellular operations, while the tightly packed chromosome acts like a secured suitcase, exclusively prepared for cell division. Here is a transverse section diagram showing the physical anatomy of that completed eukaryotic chromosome. We start on the outside with a protective layer, the pellicle, enclosing a ground substance called the matrix, which houses the main chromatids. At the center, you will notice a pinched region holding the chromatids together like a belt. This is the centromere, or the primary constriction. Attached to it is a disc-like structure called the kinetochore, which anchors the chromosome during cell division. Further down one of the arms, there is often another narrow section. This is the secondary constriction, also known as the nucleolar organizer region, or NOR. The rounded tip that extends past this pinch is the satellite body. Finally, look at the extreme tips of the chromosome arms. These ends are the telomeres, and they function exactly like the plastic aglets on your shoelaces, protecting the DNA from fraying. A precise diagram requires a clear distinction between these 2 pinch points. While the primary constriction houses the centromere, the secondary constriction is defined by its role in regenerating the nucleolus after the cell divides. Let's test that knowledge. Pause the video now, try to sketch out the 6-step word diagram, and label the major chromosome constrictions from memory. Moving to the comparative questions required by the syllabus, this table maps out 3 critical distinctions. First, autosomes control general somatic traits and make up 22 pairs in humans. Allosomes handle sex determination and consist of just a single pair. The second distinction focuses on how the material behaves. Euchromatin takes up a light stain and is transcriptionally active, meaning the cell is actively reading it. Heterochromatin stains darkly and remains inactive. The final distinction is about structural folding. Think of paranemic coiling as two parallel ropes that are easily separable. Plectonemic coiling is like two tightly braided ropes that cannot be easily pulled apart. When answering difference-based questions, biology examiners look for these specific criteria: staining properties, transcriptional activity, and the physical separability of the coils. Watch as this diagram zooms deep into the active euchromatin region of the chromosome. Here, we find the easily separable, loose structure of paranemic coiling. This physical arrangement directly serves the biological function. Loose coiling allows the cell's machinery to easily access and read active DNA. Panning into the silent heterochromatin region, the physical structure changes completely, dominated by tightly braided, inseparable plectonemic coils. Tight coiling safely locks away the sections that are currently inactive. This single graphic unifies the entire packaging process, the structural anatomy, and the core differences. Take a screenshot now and use it as your definitive revision map for the board exam.